Okay, Daniel, thank you so much.
All right, thank you, Miguel, for covering for me for a few minutes there. Okay, so I'm going to not do the presentation mode for technical reasons. Again, thank you so much for having me. I'm sort of not as low-level eye focused as many of the folks are here. So, I always admire that stuff and feel like I wish I could have impact on it, but I kind of at this higher, kind of more abstract level that has like some relationship to the biology and hopefully meaningful impact, but really curious to hear what folks think. So, in our group, we work on these three big topics, bits of them in cognitive science and neuroscience and artificial intelligence, where we basically build neural networks to solve cognitive tasks, make predictive models of brain data, and benchmark humans and machines at scale on the behaviors that we think the brain data support. And there's many relationships between these and many ways to connect them to each other. But the one that's most effective for us is this concept of hypothesis generation. It's basically using the models as a hypothesis for, what the brain might be doing. Okay, and then you can check it, and it could be wrong, it could be right, it could be partially right. In practice, what this has meant is things like optimizing neural networks for performance on some kind of ecologically reasonable task, like categorization, and then comparing to brain data. You fix all the parameters and you compare to brain data. The sort of core result here has been things like what you're seeing here, which is basically the fact that if you optimize for performance on a task, like that the brain does, the neural networks that then do the task get very strongly constrained by that optimization to look a bit like the brain. So the y-axis here is like brain likeness, essentially, right? And there's a strong correlation with the task optimized convolutional network being good model in that case at the upper right. This has gotten much more fine-grained since the picture that I showed you there, where you can separate various different parts of the brain like V1, V2, V4, posterior, central, anterior, IT. These are parts of the ventral visual pathway. Many people here are probably familiar with that. And you can separate them pretty well using the different areas of the task optimized neural network. It's a single model for all the networks, for all the areas, right? That's been carried forward to have more state-of-the-art models, which are bigger and better and fancier in various ways, and lots of work to try to characterize what makes a good model and see how well they work. And this has worked not just in vision, right? This is actually a very similar plot, but now for auditory data, actually. So word recognition performance is on the x-axis, and auditory cortex likeness is on the y-axis. And of course, the networks are different and the tasks are different, but the principle is the same. And what is that principle? Well, it's a principle of goal-driven modeling, essentially, which has sort of kind of worked both in primate and human vision, in human audition, and actually in lots of areas of mouse biology and through the work of many others in motor system, olfaction, recently in human language, right? This idea that you optimize for a task and then compare to the brain has been pretty productive at comparatively making useful productive models. But there is an underlying principle here. It's not just black box, right? It's that basically nothing in biology makes sense except in light of evolution. That's a famous quote from Theo Dobzhansky, right? Okay, so Gordon Shepard had a kind of neuroscience version, which was nothing in neuroscience makes sense except in light of behavior. What we're saying here is nothing in neuroscience really makes sense except in light of optimization, or maybe better to say nothing in computational neuroscience. Okay, so another way to restate that is the brain is highly constraining, behavior is highly constraining of the brain, right? For animals at their scale and humans, non-human primates at their scale. Okay, that's the basic principle, right? Okay, so that's all good. But there's some big problems with the types of models that I mentioned a moment ago. And maybe one of the most compelling problems is the supervision problem. So there's just no way that these animals receive labels like the ImageNet category labels, but those are critical for making the models work, right? Supervising them. This was a big problem. It's like one of the things that really was bothering me this is what kept me up at night when I started my job, okay? This is instead real visual experience looks like this. There's no labels, things are not well organized, okay? Kids see stuff like this. So for example, there's this great data set that was put together by my colleague, Mike Frank at Stanford, among others, which is basically 3 infants with a head cam tracked from age 6 months to 32 months, head mounted camera, mono, video, and audio for a couple hours a week. Okay. And so this is the type of data you would actually need to use to learn a representation if you were actually doing it the real way. Okay, and this was like a problem that I had absolutely no idea how to solve when I started my job, because of course it obviously requires some kind of unsupervised learning, not label-based learning, but they really didn't work very well, right? So there was a long history of unsupervised learning, but basically it kind of didn't work. Okay, so I mean, from a neural network point of view, this picture is very helpful to think about what had been happening, which is essentially kind of autoencoder. There's a neural network kind of like, the first part is like the encoding part, and the second part is the decoding part. And the goal of the network was to reproduce its input with some kind of penalty on the stuff in the middle, the representation in the middle. So for example, beautiful work from mid-90s showed that if you did this with a shallow network and had a sparsity penalty on the middle, essentially middle layer, then actually the filters that were learned were pretty similar to like caricatures of V1 neurons. Okay, this was sort of the beautiful sparse coding work that came from Bruno Olshausen and others, right? But the problem was that algorithms of this kind actually didn't work very well if you tried to generalize them, right? Both in terms of their ability to do tasks like categorization or position detection or size or whatever, or neural predictivity of things like V1, V4, IT. Okay, so there's this long history where it didn't work too well, and then finally, basically like in 2018, 2019, a new family of methods have arisen that have really changed things, called deep contrastive embeddings. And there's many versions of it. One of the ones that was most successful was the one developed by my former student, Cheng-Shu Zhuang. He's now at MIT. He has a whole line of amazing work on developing powerful unsupervised learning solutions, right? So the one that he developed was called local aggregation. Okay, so what did this do? I'm not going to talk about all the details. But what it basically did was, instead of trying to do an autoencoding, okay, or solve a categorization task, okay, it took the network that's represented by the DCNN there, deep convolutional neural network, and produced an embedding vector. And in this case, what the algorithm is trying to do is push together images that look like they sort of should be the same in the sense that they're clustering, and then other images in the embedding that are kind of look like they're close but not in the same cluster get pushed away, and things really far away in the embedding, nothing happens to them. So it's kind of an inverted U-shaped response function, right? And you can write that down with the math formula to make it a differentiable loss function for a neural network that you can train. But the key point is that if you do that, what ends up happening is that before training, there's kind of some structure going on. After training, you end up with multi-scale clustering of some kind that just emerges, okay? And that turned out to be, at the time, the state-of-the-art unsupervised learning solution. And it was actually better than the supervised AlexNet, right? And this was, okay, that's kind of an arbitrary point, but that was the network that started the deep learning revolution. And finally, these unsupervised things were getting better than the supervised things, the earlier version of the supervised things. So that was just to say that unsupervised learning has really started to come of age, essentially. And you can see that, you know, on tasks, it's quite a bit better. So on categorization tasks, the red bars, those are contrastive algorithms of various kinds, are much better than the earlier unsupervised learning. Not as good as something supervised on categorization. That's the black bar, of course. But when those networks are then tested on other tasks, they can actually be as good or better at transferring the networks trained on supervision, on categorization, right? So they're good at the categorization test, and they're really good at all the other tasks that the categorization network wasn't even trained for, okay? So of course, our big question at that point was, how well does it match neural data? And it turns out that there's this sort of amazing tour de force that Cheng-Shu wrote that shows that it does pretty well, okay? So actually, we're finally able to get unsupervised algorithms. This is IP neural fit here in the highest area of the ventral pathway. That networks that were as good or better than categorization-based supervised networks at solving, you know, at predicting neural response patterns. Okay, so it's sort of the first kind of qualitatively accurate unsupervised model of a higher brain area. And, you know, so this was, for us, a big advance. Of course, it doesn't solve it. There's a big gap, and we can talk about what's unsolved, of course. But just to bring it back to the type of data that it could be used on, because the networks are unsupervised, they can be made to take video data from something like the SACAM data set, that the head-mounted baby data, and push through a network of basically the same kind, although expanded for video. And it turns out that networks of this kind learn, you know, almost as well as things with like static like data sets that are like centered. So you can actually start to not only do some supervised learning, but you can have it come from natural data streams that are not curated. Okay. And this was sort of the problem that had been bedeviling me for years, and sort of actually, if you'd asked me back in 2016 when I started my lab, how long it would take us to get here, I would have said many years. Okay, but this problem has now, I don't know, it's not been solved exactly, but it's kind of come much closer to us being able to have a real candidate model for visual development. Okay, it's not perfect by any means, but it's getting better, and certainly makes more sense to investigate at this point than the categorization-based models as such. Okay, not that categorization isn't done as a downstream task, just that this is a reasonable way to think about how an organism that doesn't have that kind of label access might actually learn such a representation. So the takeaway of this part is that this contrastive and supervised approaches sort of finally have made up the supervision gap in performance and neural fits, and they kind of work okay in real data streams. But of course, they're unperfect, right? The dotted line, the neural response ceiling is not, we're not there yet. So I think there's obviously things that are missing. I'm not going to be able to tell you all of the things that are missing. I don't totally know, but I will tell you in the second part of the talk, a new thing that we've figured out that seems to put some of this in perspective. Okay, places where actually it's not just that the self-supervised models have reached parity with the supervised models, but are actually beginning to be qualitatively better and explain why that might be the right way to think about the quote loss function of the brain. Okay, so what do we know about the visual brain? Well, okay, there's at least two types of key things we know, right? We know about neuron response properties, and we know about neuron arrangement on the cortical surface, right? So neuron response properties are what I've been talking about so far, but actually neuron arrangement is one of the key things that people know, right? If you actually, this is a famous kind of picture of pinwheels in this case, I think macaque V1, okay? Right, so there's basically this neuron arrangement with essentially functional topography throughout visual cortex, right? So in early visual cortex, there's retinotopy with smooth orientation, spatial frequency maps, color response blobs, cytochrome oxidase blobs. And in higher visual cortex, there's structure basically category patches of various kinds, animacy, real world size maps, selective patches, okay? And so our goal was to think, okay, how would we connect the models that I've just described to this type of data, right? They don't make any obvious predictions about it. How are they related? Okay. It might sound like it has nothing to do with self-supervision. We're going to get back there. Okay. The core hypothesis, though, was basically there were two pieces of the loss function, and this was work that I did in collaboration with Jim DiCarlo, Colleen Equal-Spector, and folks in my group, Eshed Margalit, and others in Jim's group. And this was essentially the hypothesis that there are these two pieces of the loss function: extracting good visual features, and there's some kind of local biophysical constraint, okay? So the big picture is you train a neural network with two goals, a task performance goal and a minimization of a biophysical or spatial cost, okay? So, and then ask, do the observed things that we find emerge, like pinwheels in V1 kind of areas, patches in later areas, okay? Right, and if you look at the loss function, the idea is that this is this combination of a categorization or task-based loss function with some weight, alpha, times the spatial loss function, right? And of course, to do this, you have to embed the model in space, right? So now you have you take your deep neural network and you make it topographical by embedding the units at each layer in some kind of cortical sheet. And you can do that in a bunch of different ways. The kind of hokey way we do that now is square cortical sheets. In the future, maybe we can get more realistic cortical sheets. And then, as I say, there's this balance of loss terms applied to that network: spatial loss at all layers and task loss at the end, okay? So if you do this, how well does it do at predicting neural response data, in this case, the functional organization? And you can ask this by probing the network the way you would probe, the brain, in this case, showing it drifting gratings, if you're interested in V1, and then looking at orientation tuning maps, right? And so the answer is that it does a pretty good job. So that's actually the model. And if you zoom in, right, it's not perfectly smooth, but it's kind of smooth in the way that the Clay Reid's pictures are pre-smoothing, Okay, so not perfect, but there's pretty good structure there, and you can actually quantify exactly. Well, you can qualify and quantify it. So it's actually not just about orientation tuning, right? There's spatial frequency maps that you can measure, and then there's color preference maps that you can measure. Okay, and so these are qualitative, but of course you can quantify those things with like various quantifications. But the key point is just that on each of these different types of measures, orientation tuning, spatial frequency, color preference maps, you get things that seem to look like what is observed in macaque V1. Okay, by virtue of this kind of balance. And in some ways, what's interesting is that like very like clean models of orientation tuning that kind of build in the features. That's a little bit like the feature built-in models that you mentioned, okay, Miguel, in your talk. They're too smooth. Actually, they're like actually a little too perfect in terms of their color, in terms of the pinwheels. But the deep nets are actually closer to the actual distribution macaques, the green dotted line. Okay, so let's just yeah. So okay, so we said yeah. So qualitatively, pretty good at predicting V1-like functional organization. Of course, we were also interested in VTC. In this case, we're in humans because that's what the data is. We don't have this data in macaques, so that's VTC, kind of equivalent of IT in humans. Higher visual cortex. And so you can look at things like category selectivity and a bunch of other stuff. And if you do that, what you find is that the networks are pretty good at matching the category selectivity structure. And there's other metrics that you can use. I don't have time to go through all of them, but like things like spatial quantifications of because this is not the slideshow view, I'm getting the extra skip slides by accident. But what you can do is you can quantify things like the number of patches, the patch area, stuff like that. There's a variety of different kinds of metrics that you can use, inter-patch distances for patches of different kinds. And on all these metrics, what you find is that this kind of same network that does a pretty good job of describing V1 structure, topography, functional organization, also seems to have the higher visual cortex structure emerge, right? And so, again, it's not perfect. There's divergences of the model from the human noise ceiling, okay? But, these seem like they're able to make some progress toward making a better model there. Of course, there's always the black box question, right? And so you can ask, okay, how do you pick that apart? For me, the way to pick it apart is by looking at which components of the model are necessary for achieving match to the human data. Okay, like basically, you would call that in CS world ablation studies. Okay, so I'm going to tell you about the key ablation study that ties this work back into the first part of the talk. Okay, so I've shown you here's this total loss function. We want to figure out like why those factors matter kind of basically by doing the ablation study. So you might have thought that the loss function on the task was categorization, because that's kind of what I implied that it was, okay? And in fact, that is the one we started with, okay? But it turns out that the model that I just showed you, the one that actually is quite accurate, is the one that has self-supervision as the target, okay? The unsupervised thing from the first part of the talk, okay? If you look at comparing a categorization-based model, supervised, and the unsupervised model, it turns out that they're kind of similar in V1, but very different at the higher levels. Okay, so that's to summarize, it's actually somewhat less correct pinwheel spacing in the supervised model, and then much less good patch prediction. Okay, and there's a lot of reasons that might be interesting to figure out why that is in terms of why the self-supervised thing gives you the right distribution of units in the right locations. But it's very clear quantitatively from a bunch of case contexts that this is the case. And so in other words, for these topographical metrics, the self-supervised models are better by a substantial amount than the supervised models. And I think that really starts to give us a picture of why, why those factors might actually be there. Of course, you might want to ask directly, why do you want this kind of like spatial loss? And a good way to think about it is through wiring length. So in particular, you can measure the wiring length of the model. And it turns out that like, this is now alpha, that's that strength of the parameter of how much the spatial loss matters. So at 0, it's just basically a regular model, okay? At about the same parameter point for all of the models, for all of the metrics, that's where the optimization works best for looking like the data, and also where the wiring length is minimized. Okay, and so that's true not only across the alpha parameter, but also across all the variants of the model in terms of what components are used. So what that's really suggesting is that the loss circuits are optimized to support task transfer learning and minimize feed forward wiring length. Okay, that's what the key constraints are. And in fact, I've been talking to this in the context of the ventral pathway, and that's the work that Eshed did. But another student jointly between me and Colleen Grill-Spector have looked at this in the context of sort of larger structure of the visual system. So the kind of processing streams, right? And so of course, you're probably all familiar with there like being the parietal, lateral, ventral pathways, and like a lot of theories about why you might have these different pieces of the visual system. Okay, like what versus where, or action versus object recognition. Here's an alternative, which is basically the same thing as I showed you before. So in other words, the total loss of the system is the contrast of unsupervised loss plus a spatial loss. I put a half there to make it seem like physics, but it's, it can just pretend that it's, yeah. So the half turns out to be like the right factor in the alpha of the thing I showed you earlier, but that's probably a coincidence. Turns out that you can compare this to like the behavioral demands idea. That's kind of the classic hypothesis for the streams, which is basically, different models trained on different tasks. And you can embody that with neural networks, right? Object categorization, object detection, action detection kind of things, versus the kind of just general spatial constraints model. Self-supervised loss, having to fit into the brain and smooth it out on the cortical surface to reduce wiring length. And it turns out that if you do this, there's a lot of data here. I don't have a lot of time to talk about it in that much detail, but it turns out that you can like look at the cortical sheets that are produced that way, and the ones that come out of the self-supervised model are a much better match to the fact they basically have the pathways emerge, right? You don't have to build in 3 tasks or something. It's just basically there's units that are good for different tasks. They're present in the entire model. They get laid out and they cluster in this way that looks like the arrangement of the streams. Okay, so perhaps that can be captured simply by this learning this general visual representation, right? So just to say, well, here's the sort of summary argument. The balance of functional performance and spatial smoothness explains lots of data, like maybe a lot of what is known about the visual system. And there's really nothing vision specific about this, right? That first term is basically only vision. The self-supervised is only vision because it's applied to images. But lots of others have gone on to actually apply that to audio data, lots of other things, right? And certainly the spatial term is not vision specific either. That's like brain sheet specific. There's nothing to do with the particular system you're in, okay? So maybe there's a general theory of functional organization, right? So the speculation, the same theory might explain structure in primary auditory cortex, like, higher, or maybe even like larger structures like belt and parabelt out in the auditory areas. I don't know if people here care about that, but that's also part of the brain. And then like maybe, maybe the same theory will explain structure in things like the medioentorhinal HPC circuit, where, okay, the data input is quite different, but you could easily imagine grid cell modules emerging for basically the same reason. So the takeaways are basically contrastive unsupervised approaches have largely made up that gap. And they make a lot more sense when spatial phenomenology are taken into account, right? And there's actually this kind of unified kind of mode agnostic principle to explain a lot of data that can generate pretty fine-grained circuit inferences. So just to restate that again, I think behavior is highly constraining of the brain, but you want to also have biophysics in there too. That's what we do. And we also do cognitive science helping AI, building AI algorithms, but we'll have to talk about that at a different time. And these are the people who did the work. So thanks.
That was fascinating. We have about 1.5 minutes for questions. You guys will talk to him later.
Very nice work, Dan. I wonder how your work compares with a very rich body of work on self-organizing feature maps that has been in the literature for about, I don't know, 40 years, that has explained at least the maps in
V1, right?
V1
Yeah,
as
okay.
well as in other simpler
Right,
places
So,
by maximizing continuity and coverage.
right, so the self-organizing maps are the gray bars that are too smooth.
They are too smooth.
They're too smooth.
Oh, we need to discuss that
Yeah,
because we
they're
have written about
And
self-organizing feature
why are they
maps.
too smooth? Because you basically, to build them, you have to assume a certain set of underlying features, like orientation, selectivity, frequency, color. And the real feature space of the brain is much, much richer than that. And when you try to do smoothness on the much richer feature space,
well, that is a function of your measurement methods. How do you define the maps? If you define it at the level of a single neuron or a single synapse, you will find a lot of crudity. If you define it with a smoother, method, so this is
all right. So
this is the matter of detail as well as
not the only metrics you could use, but we tried to use the standard metrics of the field and the standard, quantitative data. And I think basically what it comes down to is this. It's hard to build a self-organizing map on the convolutional features that doesn't work so well. You can reduce the feature complexity by picking orientation tuning and select feature, and frequency and stuff. Then you end up getting things that qualitatively look very beautiful as V1-like maps, but they're just a little too smooth, basically. And to me, that's the conclusion that has emerged from it. So it's pretty close to being right. But if you actually want to have it come out of a unified model where the features are the actual rich basis of V1, which is largely not just orientation tuning cells, then you end up with having to do that through something more like this. And then it comes a little bit closer into line with the quantitative data.
Okay. This is rich for lots of discussion, but unfortunately we need to move on. Thank you so much. Our next speaker is, yes, thank you. Stand up, applause.